This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM This Week in Microbiology, episode number 319. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello, and happy new school year to everyone.
Yeah, it's September.
I'm in denial, but here it is.
Also joining us from St. Louis, Missouri, Petra Levin.
It's good to be here. It's starting to feel a little like fall.
Well, here in New York, we have multiple phases. We have a really hot August, and then all of a sudden, boom, it feels like fall, but it's fake because then it gets hot again, and then maybe in October it becomes fall.
Right.
It's really weird.
Yeah, we've got a little of that too.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. And we too are beginning to feel fall-like. It's gorgeous weather today. The humidity is gone and the breeze is out. It's about 80 beautiful degrees.
Okay, great weather for science, and specifically microbial science.
Audacious science today.
Audacious, that's good.
Audacious.
Audacious. So we're going to start with a little snippet story from Petra.
So this is actually an article from a few weeks ago from the Washington Post. And I was first drawn to it partly by the title, but also because it has a picture of an adorable calf, a baby cow, so cute, named Sushi. So I really like this article because it sort of combines microbes and global warming in a kind of positive way. The title of this article, which we have linked on the website, is "Scientists May Have Found a Radical Solution for Making Your Hamburger Less Bad for the Planet." And it's about trying to genetically engineer the cow microbiome and eliminate methane emissions.
And the journalists are Shannon Osaka and Emily Wright.
Thank you. And this was published August 25th. 2004, again in the Washington Post. And again, on the page, there's a picture of Sushi, a 4-week-old calf, at the start of a genetic engineering experiment in California. And he's just laying there, and they have a description of them sampling his microbiome. And he doesn't seem to bother when they put a tube down into him to get his microbiome out. And he's absolutely adorable.
He's very clean.
He's very clean. He lives on, I think, almond. He's like on a bed of almond or rice husks. So he's very clean. His nose is adorable and pink. Sorry, I could go on for 20 minutes about how cute this cow is. But okay, let's get to the science. So cows produce, so they have this specialized stomach called a rumen, and that's where they're able to digest things that we can't normally digest. Like they can eat sawdust, husks of things, in addition to like the grass and corn they normally eat. And the reason they're able to break down these very long polymers of like cellulose or things like this that we can't digest is because actually in the rumen, this specialized stomach, they have microbes that essentially sequentially break down these giant carbon molecules into smaller and smaller things that the cow can actually assimilate and use to grow into giant, maybe slightly less cute than Sushi cows. And if you've ever, they're ruminants, this group of organisms, because they have this extra stomach, and they also can sort of regurgitate some of this and re-chew it. So they masticate it over and over again. So you often see cows chewing, and they're called chewing their cud, or ruminating
or crud,
is the other
as
word
the
for
case
it.
may be.
Or crud. And they essentially are chewing it back up and helping the bacteria digest all of this down. And so at the end of like every degradation chain, there are often archaea, So this big domain of life that on the tree of life is sort of between the bacteria and the eukaryotes. And these are methanogens, and they can take things like molecular hydrogen or acetate or things that the cows have broken down and convert it into methane, CH4, which is then released to the environment, which is normal. This is what ruminants do.
They literally burp.
They burp it out. Yeah. But they release a lot of methane. Methane so cows produce 220 pounds of methane a year. And methane accounts for 30% of global warming, and cows are responsible for 4% of global warming. So a lot of that methane that's being produced is coming from cows. There are a lot of cows on Earth. Some are for dairy, but a lot of them are just free-range cattle that are used for meat.
And just to put that in context, 1.5 billion cattle.
Yes,
that's a lot. That's a lot
cattle.
of moos.
And the average cow makes twice as much methane or twice as many hydrocarbon emissions as your normal automobile.
Yep. In fact, ag, and probably a good portion of it from cattle because they're so large as compared to, say, chickens. I think about 20% of global warming is due to agriculture. So, and cows are a large part of that because of this methane production. So, if we can get kind of their methane production under control, we can cut down at least on that contribution to global warming. Obviously, we could all stop eating meat, but I don't think that's going to happen. And especially in wealthier countries, cattle are a really good source of food. And also cattle, because they can free range, you don't have to feed them for beef. If you go to the Midwest, if you go into Kansas or even parts of Missouri, or go down to Texas, the reason cattle are so successful is not only because people enjoy beef, but because the cows for most of their lives are just sort of out in the fields eating what's out there. So it's actually pretty practical to raise cattle.
Can I just add one other statistic that I was just struck by? They say that the average cow produces 40 gallons of saliva each day.
It's
40
called
gallons.
Slobber.
Well, it's part of this digestion,
than a
right?
Newfoundland dog.
Getting the enzymes to chew up and break down these complex carbohydrates.
Right, exactly. So that's what they're ruminating. And actually, I learned that that cows, right, can do this, but other large animals that you might sort of associate with cows, like horses, can't actually do this. They don't have the two-way valve. And in fact, horses can't even vomit, which causes lots of problems.
Mm-hmm.
But that's an aside. But cows are very good at it because it's part of their way of digesting food. And so the idea, and this is a joint project by scientists at the University of California, Davis, and also the Innovative Genomics Institute, which is an institute founded in part by Jennifer Doudna, who won the Nobel Prize for developing CRISPR, this system for gene editing, which I want to make sure everybody understands actually comes from basic research in microbiology, looking at how bacterial viruses or phages, the sort of competition between bacteria and phages, this sort of in warfare, that's where CRISPR, it's actually originally a bacterial system for preventing reinfection by certain bacterial viruses. Anyway, she had the brilliant idea to use this this system as a way to edit genes and modify the sort of genomes of different bacteria and also eukaryotic cells and all sorts of things. Anyways, so it's a joint research project by Davis, which is the big agricultural school in the University of California system, and this IGI Institute to essentially change the makeup of the cow's microbiome in the rumen in particular.
And I dropped into the show notes for your listening and viewing pleasure her TED Talk from Innovative Genomics Institute that actually describes the audacious project. And so you can learn more about CRISPR. She goes into great deal explaining CRISPR, and to have the Nobel laureate explain CRISPR to you, it's pretty cool.
Yeah, it's cool also because she's the one who developed it.
Yes,
so.
that is indeed true too.
She's the mother of
collaboration
CRISPR.
with Emmanuel.
Yes, in collaboration. She is not. She shared the Nobel Prize, and there's a third lab too. And I just love it because it just is always a good example. If somebody says, "Well, why are you doing basic research?" This is a great example of why basic
It's
research
interesting
is
that,
important.
on the other hand, another microbiologist, Pat Brown, his solution was to make impossible meat, right? Fake,
Mhm.
Right.
and said, okay, we're going to cut back on cows and we'll make fake meat. But that's harder because people don't like it as much as real beef, right?
So Pat Brown started a company called Impossible Meat.
Mhm.
And
Yep.
Impossible Meat actually relies on actually genetically engineered, I think it's soy?
Legumes.
Legumes. I think it's soy, but they produce leg hemoglobin because people figured out
Mm.
that part of why we like the way beef tastes is because of the hemoglobin from the
Mm.
blood.
Mm.
And so the leg hemoglobin, which is a plant completely independent hemoglobin, but also binds to iron to carry oxygen, is why. So, anyways, but I think it's basically a good example. I think Pat Brown's company and then eating other meats and trying to figure out different ways for people to get protein, like all together with this, the more things we can do, the better the solution. There's no
Absolutely.
one solution.
Yep.
It's multiple
I mean,
solutions.
Pat is also going after chicken and veal. He wants everything gone.
My son is vegetarian, and so, but he likes the taste of meat because he started being vegetarian when he was in third grade.
Mm.
And so we rely on these a lot. They taste pretty good. They're good.
So how are they going to solve this problem, Petra?
Okay, so the way they're solving this problem, and the reason why Sushi, the little calf, is getting his rumen sampled for microbiome is that inside the rumen are a whole bunch of bacteria, and then also these archaea from this third domain of life that are actually responsible for digesting and breaking down these kind of crazy things that the cows can eat, like sawdust or grass that normally they wouldn't be able to digest without the bacteria and the archaea. The archaea are responsible for the very last step in breaking it down, taking things like acetate, but also taking molecular hydrogen and making methane, CH4, which again is this greenhouse gas that the cows can't use, and so they burp it up. So the question is, the cows don't need the methane. They don't really need the archaea, but they do need these molecular hydrogen and things to be consumed. So the idea is, can we essentially make a probiotic, essentially something we can feed newborn calves, that will essentially seed their microbiome with bacteria and archaea that don't include the methanogens? Can we give them a microbiome that won't produce methane?
And to give you some sense of what the problem they're trying to accomplish is, they're typically about 30 different species of bacteria and archaea in the rumen of ruminants, cows, sheep, goats, whatever. And they are looking at how to engineer this 30 different species community to not make methane.
Right. So I think the thing to think about is it's very overwhelming if you think about, can we engineer in a completely new microbiome? So the tact, at least the one that they focus on in this Washington Post article, I think is very smart. So they look, and there is an organism. They have all these methanogens in them, but when they look at the microbiome, they looked for things that could do the similar reaction, that can take these byproducts of the breakdown and use them to make things, but make things that are not methane. So again, they want to sort of substitute. You can't just eliminate the archaea, but you can substitute for them. And we know actually that we can that the archaea are not essential because you can actually feed cows garlic, which I'm not sure I'd want to be near a cow that just ate enough garlic to prevent that. You can also feed them red seaweed oil,
Mm-hmm.
and
even seaweed.
And even seaweed straight. And that will essentially inhibit the growth of the methanogens. And so they're going to see what is taking the place of these guys. Obviously, it would be great if we could feed all cows these things, but it turns out that except for cows that we milk, most cows are free range, like I mentioned earlier. And so they don't really want to do that. So they're feeding sushi. So they feed sushi oil made from this red seaweed
Hence her name, or his name.
His name is Sushi. Not sure why, but it's cute.
The seaweed.
The seaweed, yes. Oh, that's right. I've read this whole thing a bunch of times. Anyways, I'm like, there's no fish involved. So anyways, sometimes it takes me a while. But anyways, they're feeding him this stuff. And then they're sampling the microbiome and looking how it's different from a cow that isn't fed the red algae. They basically want to reconfigure the cow microbiome in such a way that it resembles whatever happens when you feed them this red seaweed. And so it's still pretty early days, but they've found a bacteria whose name is,
Duodenum bacillus.
Yes, duodenum bacillus, which actually consumes the molecular hydrogen that the archaea would eat. And the idea that they have is to take this duodenum bacillus and engineer it so it grows even better, it's even more robust, and would essentially outcompete the methanogens. And so that if you can seed a newborn cow's microbiome, its rumen microbiome, with this super duodenum bacillus, that then it would outcompete the methanogens. And then the methanogens would essentially be pushed out and you would produce less methane. So that is the idea. I think it's very clever. They make this big point that they want to do something that is for all cows, that you can essentially, every cow, you could just give it this pill, right? And change its rumen microbiome at birth. So that would be easy. If the pill is cheap, it's very easy. It
like
would
a vaccine,
be very difficult.
Yeah,
exactly. Like a vaccine.
Mm-hmm.
Like a live vaccine almost. Mm-hmm. And you would do it in such a way that every cow could have this, especially when they're newborn with their mothers. And so in that way, that's the only thing you would have to do, the only intervention. And that way, the cows would go through life producing less methane because they'd have this engineered microbiome. And it might even be that they would, pass it on to their progeny, because we get our microbiomes from our parents, it turns out.
And the way the cow gets its microbiome is principally when the calf is born, the mother proceeds to lick the animal to literally inoculate it with her rumen microbiome, because after all, as Petra said, they are continuously chewing their cud. So they have a ready inoculum that is in their mouths and on their tongues. And if you've ever watched the birthing process of a cow, it's quite remarkable at how quickly the mother cleans the newborn calf, literally introducing it to her microbiome.
Right. And that's, if it wasn't cute enough already, the story,
yes,
we also have the mother cleaning the baby calf. So this is, still early days, but the idea is they're going to have to figure out a way to engineer this duodenum bacillus to outcompete the methanogens.
But that's within our reach, right?
I think
we've got decades of experience studying and engineering bacteria. So I'm optimistic.
Right. I'm optimistic too. And I'm also optimistic because they're engineering the microbiome, not the cow itself, that it will be more acceptable to people who are concerned about these things.
And this bacterium that they're trying to tinker with, Duodenobacillus, we already have in our own guts.
Oh, I didn't know that.
This is a normal microbe. As its name implies, it's associated with the duodenum or the duodenum of humans. And I dropped into the show notes a paper on it. It's a new species identified in that junction between the small intestine and the stomach.
And we've used the term audacious a number of times, in part because this is funded by a really interesting collaborative nonprofit called the Audacious Project that works with proven change makers to surface their boldest ideas for tackling global problems. So it includes more than 40 different individuals or foundations, including some ones that are familiar, like the MacArthur Foundation, the Bill and Melinda Gates Foundation, but also a number of individuals who've contributed and other groups. But they have funded projects that span a really wide range. For example, helping girls in sub-Saharan Africa thrive in school, safeguarding the ocean, by making human activity at sea visible, unlocking life-enhancing benefits for domestic migrant workers, accelerating renewable energy for a climate-secure future. So it's heartening to know that there are individuals that are really trying to push the science by providing funding, and that it's 7 years of funding they have to work on this project. So
Yeah, it's 30
outside the
million for
standard
7
model
years.
of academic research.
Right. And I like this one because, well, first of all, the microbes, but I also like it because they're trying to modify the microbiome and they're trying to do it in a way that's practical for all cows, which I think is great. They're not just trying to do one thing, and they're really going immediately for scale, which, a lot of times some of these projects are, can we make a little bit of this and then scale it up? But so that, and also I think it will be because people want to eat meat. And so you're saying, well, if you want to eat meat, we're not going to convince everybody not to eat meat. So here's another way of tackling the problem.
Or the other pushback might come from
it was creative.
The farmers, right? The people that are raised, their livelihood in the town depends on this industry. It would just modify the animal, but not the industry.
Yeah.
Well, I'm hoping that this is a sustainable technology in that once they are successful in colonizing a herd with the right bacterium and modifying the microbiome of the herd, then when new calves are born, the mothers will effectively provide the microbiome to the newborn calf. And that's probably one of their N-year aims, to see whether or not it will go vertically in the herd, whether it will move from mother to daughter.
Yeah, that would be even also great, because right now a lot of the agriculture and the sort of genetically modified specialized, like certainly in the plant sector, is one off. And you're actually, this is nonprofit, but when it's for profit, often you have the farmers have to sign things that say they won't use the seeds. So this again is I think really great research to fund, because the idea is that it will eventually hopefully be self-sustaining and available to everybody.
Yeah, and hats off to these two journalists from the Washington Post. And also they worked with their colleague Helen Ospina, who provided amazing videos and photos that are sprinkled throughout this Washington Post article. So that and the show notes description of the Audacious project, I think, are interesting reading. Join us.
And I will say that in addition to the cute cow pictures, there are some adorable graphics. Like Michelle said, totally worth
the graphics are very good. Yeah, I like them. Very accessible.
Yep.
Yes.
They're friendly, so they're
friendly. Yes.
They're cuddly graphics.
And there's some where they just have a piece of tape with handwriting, and it's just,
it's elegant. It's
very good. I like it.
You feel like somebody's talking directly to you, right?
Yeah, yeah. I must say, I like the photo. They have two cows eating, and the cows are looking right at the photographer. I think that's so cool. They're very curious, right?
They are curious. They look up if you go near them.
Yeah. All right. Thank you, Petra. Okay, I have a paper for you. I saw this, and I said I have to do this on TWiM because it's got viruses in the title and antibiotics in the title.
Great choice.
Now, what a combination. For TWiM, Nature Communications, giant viruses as reservoirs of antibiotic resistance genes. And I said, oh, wow, I didn't know this. So let's do it. This comes from South China Normal University and Sun Yat-sen University, of course, both in China. The first two co-authors, Xinyu Yi and Zhi Lang, and the senior author, Xinchan Li. And, antibiotic resistance is a problem, as you know. And how do you get antibiotic resistance? Well, it can be by mutation or horizontal gene transfer. And so in prokaryotes, that means conjugation, movement of DNA from one bacterium to another, transduction, being carried by a bacteriophage, or transformation, DNA floating around and getting into cells. So the thing that's cool here is that we know that prokaryotes exchange genetic material all the time with them, but they also occasionally get it from eukaryotes. And there is an example of this, eukaryote to prokaryote, horizontal gene transfer. a gene that conveys resistance to the antibiotic mupirocin in bacteria. There's some evidence that this came from an early evolved eukaryote. And there's been already evidence that giant viruses. Okay, so what is a giant virus? It's so it's bigger than most viruses that we understand. So they can be up to 2.3 microns in length, with genomes up to 2.5 million bases, which
Wow.
is much bigger than we think of as viruses.
That's
It's
bigger
not
than
carry-on luggage for the virus.
I
Not
think
carrying on
two
like
Staphylococcus aureus is like 2.2. So
It
this
is.
virus is bigger than the
So
Bigger
entire.
than staph.
the first giant viruses were mimi viruses, but then many more have identified, both cultured and in metagenomic data sets. And they appear to infect a wide range of organisms. And there was previous evidence that there's some genetic exchange between eukaryotes and prokaryotes mediated by these giant viruses. They seem to infect many different hosts, so eukaryotic hosts. So there have been 3 studies that report. So the term that they use here is antibiotic resistance gene, ARG. Michelle, did you remember Argif stratiatus at Columbia?
Oh, yeah.
We called him Arg.
Yeah.
I think arginine.
Arginine also, yeah. All right, so in one study, 6 giant viruses. So these giant viruses are also called nucleocytoplasmic large DNA-containing viruses because many of them reproduce in the cytosol. They set up their own shop in the cytosol, and a few of them need to communicate with the nucleus, so nucleocytoplasmic. So there is one member of a family, Marseilleviridae. They have a gene encoding dihydrofolate reductase, which, when made in yeast, it can confer resistance to certain antibiotics.
And dihydrofolate reductase is an extremely important enzyme to the bacterium because it allows it to do 1 carbon carries. So that's critical for the manufacturing of nucleic acids. And as Vincent's now going to tell us, the antibiotic that targets it.
Yeah. So, well, there's also, did I say this? I'm not sure. One of these 6 DHFR genes can confer resistance to trimethoprim and pyrimethamine in yeast. So if you take that large giant DNA virus, dihydrofolate reductase, put it in yeast, it makes them resistant to these compounds. Second study, there's a beta-lactamase gene in 21 NCLDV genomes. And this can work actually in E. coli. And then more recently, dihydrofolate reductase or beta-lactamase genes were found in 8 genomes of giant viruses reconstructed from permafrost metagenomes. Okay. So you go into places of the world that are frozen all the time. You take some ice you thaw it, and you do metagenomic, you do DNA sequencing, and you can assemble genomes of giant viruses there. One of the terms that we're going to encounter here, metagenome assembled genome. So when you take frozen ice and you sequence it, basically, that's a metagenomic experiment. You're getting all the genomes in there. And then you can computationally study it and ask, can we assemble any individual virus genomes from that? So that's why it's a metagenome assembled genome, or
Take that big
MAG.
gamish and then put it into appropriate
Yeah.
buckets.
So you have to realize you're doing this computationally. It may be wrong. Okay. The virus you put together may not exist because you've put together computationally, and there's a lot of criticism of this, but it does work sometimes, and sometimes you get, but that's different from having a cultured virus, right? A virus that you can grow in cells and culture. So here we're going to look at both. We're going to look at cells, viruses that have been grown in culture, and metagenome assembled genomes. Okay. All right. So what they did now is say, how widespread is this that we find resistance genes in these giant viral genomes? So all they do is take all the genomes that have been published, right? There's 130 actual isolates, which means somewhere there's a tube of virus that can be grown in certain hosts. So that's a virus isolate. And then 1,286 MAGs, metagenome assembled genomes. Okay. So that is not an isolate. That's computationally assembled. It may or may not exist, but they did both. And so, just to give you an idea of what viruses we're talking about, pox viruses is like, well, among the isolates, 32% are pox viruses, which would include smallpox virus, vaccinia virus, monkeypox virus. Iridoviruses are insect viruses, 10%. Mimiviridae, about 8.5%. And then the MAGs, again, Mimiviridae, 6%. Pithoviridae, another interesting giant virus, 2%. And 55% of the isolate genomes are host associated, and 90% were from of the MAGs were either from freshwater or marine environments. So, in other words, these isolates were gotten together with a host. And they also have phage. They have also done the same study with phage genomes because they would like to say, let's compare the frequency of antibiotic resistance in phage genomes. So they looked at 4,682 phage isolate and 35,595 MAGs of phages. So again, assembled genomes. And they could be assigned to 78 known phage families, of which the microviridae are the top 20%. And 58% of the isolates and 53% of the MAGs came from host-associated environments, and 28% of the MAGs were from freshwater and marine settings. So both giant viruses and bacteriophages are looked at here. So they basically searched through all of this sequence for antibiotic resistance genes or something that looks like an antibiotic resistance gene. And they find 749 ARGs in the giant viruses and 453 in phages. And they also found potential efflux pump genes in the giant viruses and phages, but they decide not to look at them any further because they may have some other functions, right? 39.5% of the giant virus genomes had some kind of antibiotic, what looked like an antibiotic resistance gene.
That was really striking.
It's a lot.
And across many different types of viruses. So this looks very widespread in nature.
And these are viruses that are not bacterial viruses. So then the question becomes, why?
Why are they carrying this luggage around?
It makes sense that a phage would.
Right. Well, these are, to be clear, these are eukaryotic genes, right?
Yeah.
Dihydrofolate reductase is a eukaryotic gene. But as we'll see, when you put these in E. coli, they work.
Right.
They work, even though they're really different, which is amazing. We'll get to that.
It's called complementation.
So, yes, 39% of the giant viruses have antibiotic ARGs, but only 1% of phages have ARGs. So the phages are less likely to carry these genes around.
Is that because they're space limited in terms of how much nucleic acid they can pack into the head?
It may be, right?
It's probably selected. They have to make a choice of how much carry-on luggage versus checked luggage they're going to actually bring with them.
So that, the many of these phages have icosahedral capsids, right? So that would be an issue. But some of these giant viruses, viruses, are icosahedral as well. So they would be limited. So I'm not sure that would explain it, right? Because it's there.
But the authors, this team, actually set out to look at the contribution of bacteriophages to antibiotic resistance transmission. And they were surprised when their initial review of literature and data is like, oh, wait, they may not be the major players here. So they pivoted then to focus on these giant viruses as well.
So the giant virus genome, overall, 0.13% of their total open reading frames could be annotated as some kind of ARG, but only 0.008% of phage genome, so much less, as we just already told you. There were 12 different kinds of antibiotic resistance genes, consisting of 19 antimicrobial gene families, and phages encoded a total of 9 antimicrobial resistance types. In the giant viruses, mostly rifampin, 51%, and trimethoprim, about 20% resistance genes. So resistance to rifampin and trimethoprim, which you can see by looking at a protein sequence. Those are the main ones. And in fact, the rifampin is mainly carried by pox viruses, and rifampin used to be used to treat pox virus infection. So probably that's not surprising. And then the trimethoprim was a major resistance gene in the Pandora viruses and the Marseille viruses. Mimi viruses tend to have mupirocin resistance, and other families had macrolide, lincosamides, streptogrammin, and peptide resistance genes in them.
Right. And these genes, these resistance genes, target antibiotics that stop basic biological processes.
Yeah.
Like the rifampin is transcription or
mRNA. mRNA.
Yeah,
transcription.
And then the trimethoprim is folate, and then you had a couple others there that target, I think, protein synthesis in the macrolides. So these are different, and they're doing very different things.
The most frequent genes are the dihydrofolate reductase resistance genes. 300 occurrences in giant viruses, 325 in phages, and these are genes that are targeted by the antibiotic trimethoprim. Okay, and then they have some other gene families, including what's called the F subtype ATP binding cassette genes, ILES genes, and glycopeptide resistance cluster associated genes, which have different mechanism. The ILES genes, for example, encode isoleucyl tRNA synthetases, which are targets of mupirocin, right? So that's the mupirocin resistance gene that they're picking up. And in bacteria, you can acquire mupirocin resistance by mutation of the ILES, the isoleucyl tRNA synthetase gene, or by acquiring a different gene, a different ILES homolog that's resistant. So these would be a candidate for that from the giant viruses if they could mix. And we'll talk about that, because you're probably wondering, well, how would these two get together?
and remember, the other thing about the unique aspect of glycopeptide antibiotics is they work after the membrane in the construction of cell wall biosynthesis as you begin to build the peptidoglycan, and they're mostly targeted against the antibiotic class like vancomycin,
right,
which is a really complicated resistance pattern because you literally have to reinvent the way the bacteria make their cell wall or their peptidoglycan.
They did a little bit of phylogenetic analysis of these genes, and they are not monophyletic. In other words, the DHFR did not start from one original DHFR gene and then evolve in different hosts. It has multiple origins, which makes sense because DHFR genes are in these organisms to begin with. So that it's not surprising that they were picked up, likely from the host, which these giant viruses use eukaryotic hosts, of course.
And I appreciated their lovely graphics, the way that they compiled all that information and made it visually accessible. So in Figure 3A, they color code each gene according to its likely origin, and they range from archaea to bacteria to eukaryotes, etc. So I'd call them shrimp plots just because they look like curled up shrimp. But
they're very nice. The third most frequently detected ORAG in these giant viruses were not present in phage genomes, and this is the ILES genes that confer resistance to mupirocin. So basically, we know that mupirocin selectively inhibits certain bacterial and archaeal transferases and ILES encoding enzymes, and they do not inhibit eukaryotic counterparts. So the eukaryotic genes, the ILES genes, are inherently mupiricin resistant. It's not like they arose in response to selection. They're there for probably another function, and they're resistant to the antibiotic. And previously, people have shown that multiple bacterial mupiricin resistant genes came from eukaryotes. So this expands that idea. And maybe the giant viruses play some role in transmitting these genes between a eukaryotic host and bacteria. All right, so then they did an experiment to ask, do any of these genes actually work? We find these genes in giant viruses. Can we put them into E. coli and will that confer resistance? So they pick two giant virus DHFR sequences. They put them in a E. coli strain that's sensitive to trimethoprim, and one is they're from two different virus families, and the proteins have similar tertiary structures. They're 33 and 36% identical with the bacterial homologs. 30% is all that these giant viruses, giant virus DFR genes are identical. Yet they both make the minimum inhibitory concentration of the E. coli from 0.5 micrograms per mil to 64.
Wow.
So they increase the amount, the MIC. So they're conferring resistance in E. coli, yet they're only 30% identical to the E. coli homolog.
But they saved the key
DHFR.
parts, I guess. I don't know.
So this gene has some function in the hosts of giant viruses. The giant viruses pick them up. I don't know why. We have no idea why they're in the giant virus genome. And if they ever got into bacteria, they would confer resistance. I mean, maybe that's the limiting step. Maybe that's not going to happen very often, but it does. Now, one other thing they looked at. So we have these genes that can potentially move around by horizontal gene transfer. There are also enzymes that are involved in mobile DNA genes moving around, like transposases and integrases, recombinases, and so forth. So they look for those, and they find them in the giant viruses in spades. Oh my gosh, 96% of the genomes of giant viruses have these mobile genetic elements in them.
Amazing.
And the nucleases are carried by 90% of the genomes. So, and the phages have way less. They have 63%. So the idea is that the giant viruses have not only potential antibiotic resistant genes, but they have the machinery to move them to different places as well. Very cool.
Wow.
Okay, so that's the story here. We have in giant viruses, 39.5% have antibiotic resistant genes. And if you're wondering what that is in bacteria overall, it's 47%. 47% of bacteria carry antibiotic resistance genes. So this is getting close. 39% is pretty darn close to
I
that.
think this really raises the question of what is an antibiotic resistance gene?
Exactly.
I mean, like, could we just take up genomes of different eukaryotes, perhaps even our own, and shotgun it into E. coli and see whether any of them, because I mean, the enzyme, as long as the enzyme is made and, processed properly in the bacteria, it's going to catalyze the reaction, I think.
Yeah,
I mean,
that's right.
The pH is right. So it's actually that's actually a good undergraduate lab idea.
Ooh. Well, I think that's, with the exception of rifampin resistance, which was probably selected by using rifampin for poxvirus infections. You say these other genes are functional in the host of the virus, the giant virus, and they happen to confer resistance in a prokaryotic host, but that's not their function in the eukaryote.
Right, but as long as they can recognize whatever the group is on the antibiotic and catalyze a reaction that destroys it, then in a way, anything could be that can have that. That's philosophically really, we're looking for antibiotic resistance genes and horizontal transfer. We're looking specifically for things that we've seen before that we know confer resistance. So
they say, well, maybe some of these genes have multiple functions, and they're not really evolved to resist antibiotics, but they have multiple functions, and that's why they can do different things when you put them in prokaryotes. So how would they get from a giant virus? So the giant virus is picking up these genes from their hosts. How would it get into a prokaryote? Dude, that's the big leap of faith here, right? Because giant viruses do not infect bacteria. I mean, there are some big viruses that infect bacteria, but that's not the NCLDVs. Those are different families of viruses.
I'm going with soil. Anything in
Mhm.
soil, and then you just need a couple of competent bacteria that can take it
Right.
up.
Picking up
Yeah,
naked
that could
DNA from a
just
giant
naked
virus
DNA.
that's
So these,
died.
yeah. Yeah, these giant viruses fall apart. Their DNA is there, and
Yeah.
it gets picked up. Yeah.
Yeah.
Exactly. Or they could get them from just any kind of DNA. I mean, again, it should be, in theory, it doesn't
Yeah.
have to come from the viruses. It could come from the host.
The other thing is it could be an accident because the mechanism of action of rifampicin in the virus is it inhibits viral assembly. The mechanism of action in rifampin in bacteria is it prevents messenger RNA synthesis. So those are two very
That's right.
different processes, and it just could be an accident that the giant virus is carrying RIF because it will allow it, if it sees it, to continue to effectively assemble itself in the presence of rifampicin, but it's an accident when the bacterium acquires the gene and says, "Oh, now I'm resistant to this thing that was preventing me from making message."
Yeah. Yeah. I
So
think,
I wonder how many accidents
right.
these things actually are reporting to us. And I don't think anyone's ever asked, what else can they do? And that's the thought experiment that Petra's saying, is how little of an
Yeah.
antibiotic resistance gene will make a protein that has activity.
This paper certainly expands kind of the classic textbook lesson about plasmids and
Oh,
bacteriophage and
No,
transposable elements
exactly.
being the main culprits in transferring antibiotic resistance. There are other sources out there.
Right. And I also wonder, I guess the other thing I wonder is like what happens when you have these things expressed like single copy on the chromosome versus on a plasmid, a high copy. I mean, again, you've got binding. Yeah. There's like so many cool questions you could ask here.
Yeah, and this is a big
So,
team, so they're equipped to ask some more big questions. It's 14 investigators, but the two
oh
lead
yes.
authors, the first is I'll describe as Jinju Li, is an associate investigator at the School of Life Sciences at South China Normal University, and they began with a bachelor's in engineering and then got a PhD in environmental microbiology and are now an associate investigator and has been quite productive, has published more than 20 papers, and primarily focusing on microbial antibiotic resistance genes and their mechanisms of transmission. And the other first author is Zhilang Lang, who is an associate professor at South China Normal University. And she is interested in a number of topics, including one kind of fascination is understanding the role of microorganisms during ecological restoration of mine wasteland and its associated acidic environment. But she also collaborates on this project looking at antibiotic resistance in microorganisms.
so one last comment on where this transfer from eukaryote to prokaryote. They like amoeba because amoeba are infected by some of these giant viruses, and they also take up bacteria, right? They eat them, and so they're going to be in the same cell, and maybe all these mobile genetic elements in the giant virus genomes can facilitate the movement of these genes, not just these the ones that we're talking about here, the antibiotic resistance, but other genes as well. Obviously, it's not going to be selective,
Yeah.
and so they say the presence of so many giant viruses with both antibiotic resistance genes and mobile genetic. The other thing we didn't mention, they also find virulence factors in these giant virus genomes. So virulence factors that are known in bacteria to change virulence. You can see homologs of those in in the giant viruses. And so they're worried if those get transferred over with the antibiotic resistance, that could be a bad situation there. So
a riff on pathogenicity islands, not from bacteria, but from giant viruses.
yeah, very
Yeah.
interesting. So,
And the viruses, the large viruses that did have virulence factors in their genomes were more likely to have these antibiotic resistance genes as well.
right.
Deadly combination.
So they say we should be paying more attention to this. Probably a good idea, right?
Yeah.
Another case where basic science is important.
Oh, yes. Very important. And so I think this was published in August. So this is going to be noticed by people. Oh, we have to pay some attention to this. So you can imagine many more studies that would be done. For example, I'm thinking if you know a certain distribution of giant viruses or their hosts, you can make correlations where antimicrobial resistance arises and so forth. And that's never been done. So I think that would be pretty cool. All right, that is TWiM 319. You can find show notes at microbe.tv/TWiM. And if you want to send in a question or comment, TWiM@microbe.tv. If you enjoy these discussions where we try and not only teach you science, but get you excited about it, we'd love to have your support. Go to microbe.tv/contribute. Michelle Swanson's at the University of Michigan. Thank you, Michelle.
Thank you, and go blue.
Go blue. It's that time of year.
Yes.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, everyone.
And Petra Levins at Washington University. Thanks, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.